I think the thing that you're most well known for is that you were the first to lead the effort to sequence the human genome.
So all of medicine today is symptomatic medicine.
Yeah, sick care.
It's sick care. You have to be sick to be seen. What we show is that diseases take time to develop. By the time they're detectable and you're sick, it's quite often too late. Almost all the tumors we've discovered in screening close to 7,000 people have all been treatable and curable.
Wow.
Because, but the wave actually broke over the boat, and there was a while, all that was sticking out was my head, just at neck level, and the mast. And all I could see was boiling water all around me. And I was
my gosh.
hanging on very tight to the wheel.
Dr. Craig Ventor, what a pleasure to have you here with me today. I am just so honored to have you. You've got such an incredible background, such an amazing history, one of the top scientists in the world, listed as one of the most influential people in the world in 2010, and an author of an incredible series of books. One is, that we're going to talk about both of them actually, but the most recent one is The Voyage of the Sorcerer 2. And that is an exciting story. But I think the thing that you're most well known for is that you were the first to lead the effort to sequence the human genome. And I remember when that news came out back in the 90s. It was kind of like, I knew you were working on it, and I heard your name even back then. I think it was probably around 1995 when I first heard your name.
That was when we did the very first genome. It was a bacteria. So,
yeah, bacteria.
Yeah.
And then you went on to do the human genome. Yeah. When was that, like 98?
99.
99.
And it was announced at the White House in 2000.
Yeah. That was a momentous moment. So can you tell me a little bit about how you got to this place? And you have this incredible institute. One is in San Diego, another was in Maryland as well. And I mean, what an incredible career you've had, and what an honor to have you here with me.
Well, thanks for having me.
We had such a great conversation yesterday, too. Very broad.
It was so enjoyable, I couldn't say no. So it's
absolutely.
So I'm pleased to be here, and give me a chance to get out and fly up here today. So it's
yeah. You flew your plane up here too, which was really commendable, especially with as much air traffic as there's been in the last day.
Had to dodge a few big jets coming in.
Yeah, good for you.
They said, don't turn yet. Wait till that jet gets out of here.
Well, you happen to have the one kind of plane in the world that I would probably want to maybe own one day, which is the one that has its own parachute system, which is pretty cool. I think that's a brilliant system. I'm surprised more planes don't have that, actually.
It's really fantastic. Unfortunately, I haven't had to use it yet.
Well, I'd never gone forward to get my license, but I did learn to fly. I did it for several hours, but to go forward with the whole license process was quite a thing to do. And you did it at the age of 72.
That's right. I got my pilot's license at 72, thanks to COVID primarily. It gave me a break from traveling around the world every week. And so I thought, I have this clear period of quiet time. So I bought a plane and just learned to fly it.
Wow. So tell me, how did it feel back in 95 and then 98 and then the announcement in 2000 of mapping the human genome? And what a fascinating story, building this computer that you said was gigantic and cost something like $100 million just to put in place, just to be able to do the mapping of it.
Well, all the things were incredible, but when you're a scientist, you're doing things primarily for the science. So when we sequenced the first genome in history, we were just trying a new method that I had developed. We didn't set out to sequence the first one in history because we didn't know if the method would work. But it ended up being years before anybody else with government programs could even approach. And as we talked yesterday, it uses primarily new computer algorithms, and a lot of unique mathematics goes into it, and but it felt fantastic when we achieved it, and then knew we ended up being first in the world. And the same with the human genome of, it was just unbelievable. As I wrote in my book, there were probably 1,000 reasons our experiment should have failed. because it was new technology, new algorithms, a new computer, and it all came together because of fantastic people that made everything work.
Now, when you kind of look on your life, how would you describe it? What would be the word that you would use to describe your life story?
Probably incredible. I would have been voted the least likely person to ever become a successful scientist in high school.
Well, why is that?
Well, because I almost flunked out of high school.
There's hope. There's hope for everyone.
No, I think my book actually in the story gives hope to a lot of parents about their kids. I had what might be described as a learning disability, aphantasia, where I don't see any pictures at all if I close my eyes. So everything I learn is conceptual. So rote memorization is a difficult task, and our education system is built on that. So I just rebelled against that. I took part in sports. I built boats, had lots of girlfriends, had a lot of fun, but almost flunked out. I got a D- instead of an F in a government class because I wrote a paper knowing the instructor. This was back when Barry Goldwater was running for president.
Okay, this is a while back.
Yes, so
When was that?
1964.
64, yeah.
And even though I didn't believe it, I wrote a paper of why Goldwater should be president. And so I got a D- instead of an F in the class. So otherwise, I wouldn't have graduated high school.
What if you'd picked the other candidate?
Yeah, I would have flunked for sure.
So that would have been against Lyndon Johnson.
Yeah. And I moved to Southern California to take up a surfing career. I was offered a swimming scholarship to Arizona State.
Really?
But I didn't want to just depend on swimming 5 hours a day for the next 4 years, and I wasn't sure if I was ready for college. So I moved down here to Newport Beach and took up surfing, body surfing at the Wedge. And the only drawback was I got drafted off the beach. I say I got drafted off my surfboard here because 1965.
So this is when you were living in Newport, and
Yeah.
you were paying exorbitant rent, right?
Yeah,
Like $100 a month or
$100
something.
a month. It was amazing. Couldn't even do that a day now, probably. So it's getting drafted was probably a changing point in my life. It ended up being a good thing. I'm one of the few people that came out of a Vietnam experience with a really positive attitude and positive outcome, where it was in part because of my job. I was a corpsman, so my job was saving lives. But for quite a while, I was a doctor for a orphanage in a Vietnamese village. I found it just incredibly rewarding to do. I was doing major surgery. I turned 21 in Vietnam, but in the military, you can do whatever your skill set allows you to do and what authority doctors give you. I was a very, very quick learner. At 19, Balboa Hospital, I was teaching interns and residents how to do spinal taps and liver biopsies.
Wow.
Because there's no malpractice for medicine in the military. So I had great teachers, physicians I worked with. And so I just learned so much. I probably, people say that I experienced more practice of medicine 1 year in Vietnam than most trauma surgeons experience in their lives. Not something I would care to repeat.
Yeah.
But you learn what's important in life, and you learn what keeps people alive, and part of it is their mental attitude. Some people give up and die. Some want to live and overcome amazing deficiencies in what's left of their body. So it was, but I came out of it wanting to become a physician and practice third world medicine. I had to start at a community college in Northern California because I was such a poor student. I couldn't get into the university, but we have such a fantastic system in this state. If you take transfer classes at a community college and you get a B average or better, UC has to take you as a transfer student.
Something isn't right. The ease of modern digital communication comes with costs. What are these colossal technology companies doing with our words, with our energy, and with our very beings? Can you imagine an event horizon where they use against us what we unknowingly gave? Orion emerges as an end-to-end quantum secure messenger that places the freedom of expression back where it belongs, with the people. More than a platform, it's a movement to reclaim your voice and freedom, a movement of people with profound ideas and a deep desire to help the human race. Let's get back to communicating meaningfully in our lives. It's time to rise above the surveillance, the shadow bans, and the censorship. It's time to free our voices. In a time when free speech and assembly are absolutely necessary, Orion stands tall, ensuring that you can free your voice.
And so it's fantastic. It's a way for people to afford the first 2 years of college for a tiny fraction of going to a university. And my education was as good or better than people get the first 2 years at a major campus. But when I transferred to San Diego, I was introduced to high-end science for the first time. I'd never experienced it before, didn't even know it really existed. And I was just fascinated by it. I made a major discovery as an undergraduate, published my first research paper, and just, was captured. And so,
what was that regarding?
Well, I've always been an adrenaline junkie, so I was studying adrenaline. And there was a big debate of whether adrenaline worked on the outside or the inside of cells. And I attached adrenaline chemically to tiny glass beads so they couldn't enter the cell, and proved that it worked on the outside of the cell. So it was a very innovative approach and got a lot of attention for me. So I have the shortest PhD on record for the UC system, and I went immediately into a medical school professor position. So I never did a postdoc or anything else. So
wow,
I kind of made up for lost time. I think because of the Vietnam experience.
Yeah. Also, it reminds me. I used to live in Australia, and I worked in the cardiology field, and I used to be a cardiac tech for a while. And when we would go in and implant an implantable cardio defibrillator, I would have to be the one who pushed the buttons. In Australia, you could do this. Yeah. I pushed the induced ventricular tachycardia and induced ventricular fibrillation so that you'd actually induce a heart attack so we could test to see if the implant was working, which is kind of terrifying for the patients. Imagine they're like, okay, just lay down. Usually some old lady or some older gentleman. And you're like, okay, we're going to just do something here to
we're this. We're going to turn off your heart for a moment.
We're going to turn off your heart for a moment. It'll be fine, really. Yeah. And I used to do that. And I would go into all the open heart surgeries. And one of the things I noticed about the system, which I think is far superior, candidly, to our system here in the United States, is that at a much younger age, because they have tort reform already in place, they have limits on how much you can basically sue for. At a much younger age, first of all, you go straight into medical school. Yeah. You don't do your undergraduate, so 4 years is saved.
And it's usually a 6-year straight through.
That's program. A 6-year straight through program. And so they have people that are at the end of their med school that are actually doing surgeries. Yeah. Whereas you're probably at least another 4 to 6 years in the U.S. system before you could actually do that. Yeah. And I think it increases dramatically the amount of debt for doctors going into, the practice of medicine. And largely it's related to the lack of tort reform.
Well, and as military medicine shows, and now physicians' assistants, probably 98% of medicine can be practiced by highly skilled technicians. Yeah. And so in Vietnam, I would do independent surgery because they trusted me that if I saw something unusual, I'd go get help. But almost everything that I found was unusual, they didn't know what it was either. And so it's based on a trust and a skill set system, not based on how many years you were in college or what your degrees were. It's like, can you do this and do it extremely well? And because I could work extremely well with my hands, I was able to do extraordinary things, but so were thousands and thousands of others. You know, the special forces medics, the SEAL medics. I mean, they're truly extraordinary individuals.
Yeah. They have so much experience, real life field
Yes.
experience. It's not didactic.
Right.
I think we spend so much time on the didactic now. So when you were in university, though, I'm sure there probably wasn't even a specialty that you would consider yourself in today. You know, this very, very highly specified genomic or molecular biology to the degree of specificity.
There was a molecular biology class that I had to take as part of my major, but it was primitive history of what happened in the 1940s and the 50s. The word genome probably didn't even exist yet, so I certainly wasn't studying that. And I trained with some of the best biochemists in the world, and it was when you train with the top scientists in the world, you learn a different approach. And it fit with what I came out of Vietnam with, of not being afraid to take risks, because I learned the main thing I had to lose was my life.
Well, that's a pretty significant learning.
Getting through that and not losing that on multiple occasions, I was not afraid to take risk. And my mentor, the late Nate Kaplan, one of the world's famous biochemists, said the biggest mistake most scientists make, and I think it applies to general life as well, people talk themselves out of doing the experiment.
Oh, well, that's probably true with great entrepreneurial ideas and everything. I think self-doubt probably has destroyed more things than governments or competition ever has, as far as new ideas that could change the world.
And I often get labeled as having a huge ego, but I'm not even sure what all that extends to, other than if you don't believe in yourself and your own ideas, you'll never accomplish anything.
Yeah.
They don't have to be at other people's expense, but sometimes new ideas fracture a lot of old ones, and people get very attached to their old ideas. So it gets very political, even in science.
Oh, for sure. How have you changed over the years? Like, how has your approach or look at life changed since the time you were a young whippersnapper, coming up with this new research about adrenaline, whether it goes inside or outside?
Well, it's certainly a lot more aggressive and a lot more energetic. So in the 3 years,
you're still pretty energetic.
Yeah, in the 3 years I was in graduate school, I published 21 major papers.
Wow.
Can you imagine over the next 3 years writing 21 major papers today? But you know, I've taken on some of the biggest projects. And the amazing thing is I've been right like 99% of the time with the ideas. Lots of little ideas fail. You know, we all face failure in life. I've been married 3 times. It took me that to learn how to get it right. So I'm a slow learner in some fields, but fast in others. But I'm probably, even though I keep it well hidden, I'm probably a lot more mellow now than I used to be.
You know, one thing I've noticed for myself, at least, is I think when I was 15, I used to think my parents weren't very smart. And then because they couldn't get the right decisions on things, it seemed black and white to me. Then by the time, like Mark Twain said, by the time I was 22, I was amazed at how much more genius my parents had become in 7 years, only 7 years. And then every year thereafter, they just seemed to get smarter and smarter. And everything that I thought was black and white turned into shades of gray, and then actually a full spectrum of color as, you know, the older I got. So I definitely have softened my own views on things. I believe I can learn something from everyone now, and I don't think I felt necessarily that way when I was very young.
It's when I turned 75, I decided to get much more choosy of what I did with my time and who I spent my time with. And I've put much more into my own life. I've always been so busy, you know, as I say, you never have time to stop and smell the roses. You know, these are all major multi-year sprints. You're literally working around the clock to accomplish something, and you know, but the goals are so worthwhile. But you miss a lot of life in the process, and so So I'm actually enjoying life a lot more now, even though, as you've seen, I've sailed around the world in my own boat. You know, I learned to fly. I do all kinds of things, but I try to mix them with science and things that I want to accomplish.
Do you look back and did you have like any major mentors along the way?
Everybody that's successful has had really good people help them out at different stages. In the military, the short time I was there, I got sent to the brig twice.
You were definitely a rebel.
And in Vietnam, I was sent there for an altercation with an officer, and a physician, Ron Nadell, knew about what a good reputation I had as a corpsman and a technician, and saved me and took me into his clinic and trained me. So he taught me a lot, and he's still a friend to this day.
Wow.
And he said, the only deal is if you get the Nobel Prize, you have to take me with you, because I'm responsible that you're here. So, but I've had a few really great teachers, particularly when I started back in college after the military, because I had, basically a failed response earlier with the education system, so I was a little terrified to start it. And I had this phenomenal English teacher, Bruce Cameron, who every assignment he gave, I didn't do it. I rewrote the assignment for something I wanted to do, but fortunately I did it better in his eyes than what his assignment was. So he was really fascinated by that and became a friend and mentor for the rest of his life. My mentor when I was getting my PhD, Nate Kaplan, really took me under his wing. People were fascinated with my corpsman and Vietnam background.
Well, both your parents were in the Marine Corps too,
They
right?
met in the Marine Corps, and my older brother was born at a Marine base. Fortunately, I came after that.
Wow. So it does sound like you've had mentors along the way that have had a pretty big impact. I could just tell by the emotion I even felt just when you were saying that. And I mean, as you kind of look back on that lifetime of incredible accomplishment, there had to be moments where it seemed like you might not succeed at whatever it was that you were trying to do.
There's been lots of those moments.
Can you tell me about the most significant of those?
Well, the most recently, a reporter from Nature was interviewing me about my book about sailing around the world. And she said, it seems like you had a lot of near-death experiences. And I said, yes, but they weren't planned to be that.
Well, yeah, hope not.
So I've had a lot of those in life, and have, we talked about racing cars. And
I've had a few
things.
myself. I've probably had, I could think of probably 6 times that I've had truly near-death experiences. One was being chased by 30 elephants on a wet horse that had no saddle.
Wow.
And they were only 15 feet behind me.
Wow.
At full speed. That was pretty scary. After I just crossed a river in the Okavango Delta in Africa. And I was actually there with Dan Quayle. He happened to be on the same trip. And we all got chased by this herd of elephants. So that was scary. I've been in 3 plane crashes, which is pretty terrifying. One was a plane, Delta Flight 88, flying in from Singapore to LAX and had no landing gear. So we did, the belly pop on the front on the front end of the plane. It's actually scarier than it sounds. I mean, you would think, okay, you can, because it had back landing gear, it just didn't have the front.
No, quite often those lead to fires. And
we had fire. We definitely had fire.
When you're a giant fuel tank flying through the air.
Oh, we went off the ditch. We went off the end of the runway. We were in an MD-11. It's a pretty big plane.
Yeah.
And that plane famously had a problem with the front landing gear.
They had a problem of engines falling off too. So.
Yeah, that's a small issue.
Yeah.
But, progress. What were the near-death experiences you had?
I mean, it's everything from being in huge storms out in the middle of the ocean. People now see a lot of videos on Instagram that, without being out there, you couldn't imagine how rough and terrifying it can be. We're off of Portugal, where they have some of the largest waves in the world.
That's where the surfers go.
Yeah, just three of us on a 100-foot
Is it Navarre
boat.
or something? Yeah.
And but big storms develop offshore. It was a rogue wave, probably around 100 foot.
Wow.
And literally surfing down with a 100-foot long sailboat. And with this, if you can't hold it in the wave, unfortunately I had a surfing background, so it helped a lot. You just get rolled over and
capsize.
That'd be it. So, but the wave actually broke over the boat, and there was a while, all that was sticking out was my head, just at neck level, and the mast. And all I could see was boiling water all around me. And I was
my gosh.
hanging on very tight to the wheel. But I entered the transatlantic race, and I actually won the contemporary division. It was the only ocean race I ever entered. And it's because the boat I had could surf down these waves, and the other big boats couldn't. We had 3 major gales going across. So what became a frightening thing became a skill set that I kind of mastered. But it's all these things, you're very much on the edge, and you can only stay at the helm 30 minutes at a time because it's so intense. Because if you lose concentration for a second, you're screwed. So, I had a girlfriend's father when I was in high school threatened to shoot me in the head for climbing in a window.
I've had that too. We share that experience, I think. And I lived in Nebraska at the time, which apparently there's an old law in the book that if you find a stranger in your house, you can shoot to kill.
Yeah. Well, I was not a stranger, but I shouldn't have been in that certain room.
Yeah, I think that's exactly the experience I had too. I was too young. But okay, but if you go back to like 1998, I mean, there was a government program that was very well funded.
Yeah, they had
to go after the sequencing.
$5 billion. We had $300 million. So they're both big numbers. One's very much bigger than the other. But like government work projects, they had a distributed labor program. Some people joked early on you should make prisoners sequence the genome because it's so tedious. And they broke it down into lots of projects and distributed it around the world. Whereas my approach deals with it all at once and depends on mathematics and computing to reassemble everything. So just very, very different approaches. But they weren't exactly friendly. They didn't expect anybody to ever enter their domain with their $5 billion federal program.
So this was an NIH-driven program.
Yes. So the only parallel that people born later might see is what Elon Musk has done with SpaceX.
Versus NASA.
And now they're working together, but private companies with smart individuals and great teams can accomplish a lot more than the government can, in part just because they have the freedom to go faster and do things that a giant bureaucracy keeps you from doing.
So I've always kind of believed in this, I would call it an axiom for myself, which has been the only obstacles that are real are the ones that we continue to allow ourselves to imagine and believe in. And I think maybe that's why bureaucracies don't do as well, because it's not as much on somebody's willpower to kind of fight through those difficult moments. It's like there must be an entrepreneur and a manager in companies. I work with both, and I've been both. And An entrepreneur, they've got to basically put the food on the table and they've got to make sure the money's there for the company. Whereas the manager's like, it's like the difference between the chicken and the pig in making breakfast, which we've probably heard that one before, which is, the difference between the chicken and the pig is the chicken is involved, but the pig is committed, right? If you're going to be having eggs and bacon. Yeah. And that's the difference, I think. The private companies that have to deal with 300 million.
Contributions to make. So.
Yes, that's right. So it's definitely everyone has their part to play, but there's that having to believe and see things through that really makes the difference. Just like you holding on to your wheel as the water's all the way up to your neck. I'm sure you had to think for a moment, this could end badly.
Yeah. Well, and I thought that all the time with sequencing the human genome. It would have, because it was such a public race, everybody was just so fascinated by getting this information that if my method failed, it would have been the most spectacular flame out in science history.
There's no prize for second place in this one.
There was no room for second place, although the government created room for themselves.
Yes, well, that's different.
and they called it first. But what I found is when you're a leader and you truly believe in an idea and approach, and there's very narrow lines, there's reality and there's delusions of grandeur.
Yeah.
And you have to make sure when you have what other people consider a wild, undoable idea, that there's a really sound basis for it. And that's where my aphantasia and my understanding of complex systems, I can put things together that don't seem obvious to other people even when it's laid out in front of them. But the extraordinary thing that happened is I formed 4 teams. We had an algorithm team of top mathematicians to write this new algorithm.
I was just going to ask, how many mathematicians did you have and computer scientists?
Yeah, we had a lot of computer scientists because we had a team to build this new $150 million computer. It was the 3rd largest civilian computer in the world at the time. It was 1.5 teraflops. And now you can get a laptop that does that. So fortunately, computing has changed a lot. But in 1999, there was nothing. And this was all based on the alpha chip and putting thousands of them together to be able to run the calculations to assemble the human genome. And so the computer could have failed. The algorithms could have failed. We had a team that had to set up robotics to make the 25 million fragments of DNA in a pure form that they could go on the sequencing machines. We had 350 of these roughly $350,000 instruments that were made specifically. And the company that made them is the one that put up the $300 million for me. They thought their machine would work well with my method. But it had a robotic arm, and early on testing them, sometimes the arm would fly across the room. We're lucky people didn't get killed. But I put people that I trusted and believed in charge of each of these 4 units, and they all outperformed even their best imagination of what they thought was possible. They inspired people on their teams to do that. I inspired all of them to do it. And that's why it worked, was because everybody put out at 150% level because they believed in it. They knew where they were making history. And if they put in the effort, they had a chance to make it real. And that doesn't happen with federal programs. That doesn't happen with make work programs and distributed things for the, raising the science economy. It has to be based on people and them believing in themselves and inspiring others. And so it was the most extraordinary team. And my friend and Nobel laureate, Ham Smith, who just turned 94, who helped make all this possible, he said the problem is we did it too quickly. We didn't get a chance to enjoy it because we were done in a year and a half.
Yeah, that's very fast.
And it was supposed to be a 15-year government project.
Wow. Wow. So,
But when it worked, it was just unbelievably fantastic. And until the computer run stopped, we had no idea if it worked or not. And so the White House was calling me every day, because they were scheduling the White House event on when our computer actually spit out the human genome. It had nothing to do, the public effort didn't have anything at the time. They were just going along and claiming it was a tie. So the whole event was driven by when our computer at Solera actually stopped running and whether it was a genome or not. So it was all scheduled, and they would call every day, basically, is it soup yet? And it was, we didn't know until the run stopped. It took several weeks of this computer running around the clock.
I remember seeing when I was in MBA school, I would see Solera stock quotes all the time.
Yeah.
And people were talking about it all the time. And you raised something like $1 billion to do this.
Before the latest rounds of companies, I had the largest raise on Wall Street. Less than 5% of a company with no revenue raised $1 billion cash. People were just so excited about what they were doing. They wanted to participate, so they just kept buying the stock. We weren't pushing it. We got up to a $25 billion market cap.
Wow.
In a year and a half. And just before this public raise, the stock was at 500, and they decided it was too high, so it got split to 250. The next day, it was back at 500.
Wow.
I wish we could do that again.
That'd be nice. So, but you did something around, after that time, as you were still CEO of Solera, and you actually donated a very large percentage of your ownership to endowing, effectively, the Institute.
Yes. So that's my goal of sequencing the human genome was to do it and to make history. The fact that it was a company that had very valuable stock, I gave that all to my not-for-profit research institute. And while I was CEO, I could not sell. I was locked up. But my institute could, as long as I didn't take part in the decision to sell or not. And so they sold stock, raised over $200 million. And that became the endowment that's kept us going for the last 32 years.
Wow.
So it's paid back in much nicer ways than having a big bank account.
That's incredible. So what do you think is next for this whole field?
Well, the problem 25 years after we sequenced the human genome, we're still trying to understand it. We're still at the 1 or 2% level at best. So that's why I started the company Human Longevity, to instead of just sequencing genomes, which is what the government does, to measure all the physical characteristics of a person so it could be compared to their genome.
Mm-hmm.
And my view is that's the only way we're going to truly understand our own genetic code. But as a part of doing that, and we started just screening healthy people with a very intensive, non-invasive protocol, including whole body MRI, echocardiogram, numerous tests. We discovered roughly 50% of so-called healthy people had a major disease that they were unaware of. 5% of everybody over 50 had a major tumor that they didn't know they had, including myself and Ham Smith. Setting up the testing saved both of our lives.
He as well, really?
Yes.
Wow, I didn't know that.
And so after I went through and testing the machine, discovered that I had high-grade prostate cancer after being told by the top medical system that I didn't have it. I paid for all my employees at the Ventor Institute to go through, and Ham was one of them, and we discovered that he had a major lung lymphoma. Just even on the MRI, it looked like an angry fire growing in his lungs. He had radiation treatment right away. It knocked down the tumor. His physician said if we had not discovered it, he would have been dead in 6 weeks.
So this is a newer type of technology that's used in MRI.
It's totally new technology of it's mathematically post-processing the MRI data, and it makes tumors light up like light bulbs. But there's, like everything new, there's tremendous resistance to using it. And the resistance stems back to the 1970s when CT scanners came out, and they were a major invention. Everybody was excited because, you get whole body CT scans. But CT, being X-ray, can't distinguish between a tumor and a cyst. And so they developed this whole field called incidentalomas, where people had to go and get follow-up workup, sometimes biopsies, to work out was it a cyst or a tumor. Insurance companies balked at paying for all these tests, and it developed into an ethos of it was unethical to do CT scans on apparently healthy people. So all of medicine today is symptomatic medicine.
Yeah, it's sick care.
It's sick care. You have to be sick to be seen. What we show is that diseases take time to develop. By the time they're detectable and you're sick, it's quite often too late. Almost all the tumors we've discovered in screening close to 7,000 people have all been treatable and curable.
Wow.
Because they were found at an early stage before people had any symptoms. We found 1% of the population have brain aneurysms. So the new MRI techniques, all the blood vessels in your brain just show up like a beautiful map of your brain. And aneurysms, you just see a little
So with
bubble.
an aneurysm, is it like cancer with this particular new type of specific MRI technology? Does it have a similar signature where the nucleus has a larger diameter?
No, actually, the aneurysm,
and the water inside is different?
It just looks like, when you have a bleb on an inner tube, when you get a blowout, that's the same as an aneurysm. It's literally a bubble that forms. So you just see in the MRI picture of the blood vessels, you actually see the bleb, the bubble sticking out. And so they're readily detectable.
So that could be done with more of a standard MRI, I'm guessing, then. Than maybe this new.
If it's done with contrast media.
Yeah.
So our post-processing does help that show up. But they're treatable in 15 minutes with a catheter as an outpatient.
Wow.
But the data was skewed. We didn't find very many people over 60 with brain aneurysms.
Because they're
That
probably
means
not
they already
alive.
died.
Yeah.
And so just with that alone, you could prevent deaths of 1% of the population.
Wow. So, but in the cancer case, and you're a cancer survivor because of this technology, and what it was able to do was to identify this change in the nucleus that then also leads to the water being inside the membrane of the nucleus being less mobile.
That's right.
And so more gel-like, I'm guessing.
Yeah, and so that gives off a different magnetic resonance signal in the MRI. It's a signal that causes the tumors to actually, they light up almost like a white light bulb on the MRI.
Which is fascinating for me because I'm now very interested to see if I could figure out a way to measure the dihedral angle of that gel-like water that's less mobile. Because I think it is probably a different state of water.
There's probably single cell technologies that would allow that, but that's beyond my expertise.
So what do you think's next? What do you think about gene editing and CRISPR and phenotypic expression and being able to modulate things like that? What are your thoughts?
Well, we're working right now to greatly expand this pre-symptomatic testing. My calculation is it could save the federal government $2 trillion a year in medical bills to detect things early and treat them before they develop into major disease. I mean, in my case, my tumors were caught just before they metastasized. So it was simply a matter of removing my prostate. If we'd waited 6 weeks, that probably would have metastasized, and then we'd had to, get radiation and go through all these treatments that are expensive. And with the type of tumor I have, the lifespan is usually less than 5 years.
Wow.
So I was happy when I got past the 5-year mark post-surgery, and
congratulations.
No returns. And but we've done that now for a lot of people. It was rewarding that the system I developed saved my own life just testing it. It was quite a surprise and a shock. It's not at all what I expected, but I'm alive today. We talked about risk of death.
Heart disease.
I'm alive today because of modern medicine. My father died at age 59 from sudden cardiac death, from just having all the things. He smoked, ate a high cholesterol diet. He was a golfer. He got some exercise and stuff, but smoking is the number one killer on its own. But the techniques we have, it's one of the frustrations I have, would have easily saved his life had we had them back then. But unfortunately we didn't. But those are the kinds of things that motivate you to change your own life. We all look to our family history for hints of things that affect our own longevity and do something about it. So, I'm on anti-cholesterol medication. I have well-controlled blood pressure. I don't smoke. Unfortunately, I hope my cardiologist isn't listening, but I still have a vodka martini fairly frequently.
We'll have to have one sometime.
But I read, they now say any type of alcohol is bad for you, but a new study came out I like. So you cherry pick the studies you like. It's like if having a drink in the evening relaxes you, that's actually good for your cardiac health. And having a martini every night actually helps me settle down.
There you go.
So even a good scientist can rationalize behavior.
That's right. That's exactly right. So that's interesting. Yeah, definitely the smoking thing. I can imagine for people that do have that, ischemia, occlusions, all kinds of issues that they're going to be facing. Does your MRI system that you do at Human Longevity for people, I guess it captures all of that?
Well, it's less
like a full body scan.
Useful with soft tissue like lung or colon cancer, so we do it without contrast. Because contrast is invasive, a certain percentage of people die when you give them contrast. And for the kind of clinical tests we were doing, of trying to get data on this, we didn't want to put people at any risk at all. So there are added techniques you can add to the MRI by adding contrast media that detects breast and colon and lung cancer. CT scans are very good at detecting soft tissue diseases, and colonoscopy is very effective. Mammography is sort of back where CT scanning was. It's X-ray, so it can't distinguish cysts from a tumor.
Yeah.
And so women that have those have to get follow-up care. So we're trying to see if our techniques...
And often if they're going to go in for a biopsy, they're going to go ahead and do the lumpectomy or a mastectomy if they've got the markers.
Well, so the genetic markers like BRCA1 and BRCA2, there's lots of misinformation about them. Physicians frequently tell women that if they have those genetic changes, that they're at a 90% plus risk for breast or ovarian cancer. Well, it turns out that's not true unless you have a family history. Essentially, every woman in your family has breast or ovarian cancer. Without that family history, those tests give you a 50% lifetime risk of getting breast or ovarian cancer, which means BRCA1 and BRCA2 are not the cause of breast or ovarian cancer. There are markers related to it. And
It's not causal, but it could be corollary.
Yeah. But the fact that the family history is so important, it means we still don't know which genes are actually the causes of breast and ovarian cancer yet.
Interesting. So in your own case, I'm sure you went back and looked at your sequencing after you got the diagnosis on your prostate cancer.
Yeah.
And what did you find? Can I ask?
Well, yeah, no, it was really fascinating because I didn't have any obvious tumor markers, even though I have a high rate of skin cancer, but, dermatology surgeon really, I've had, I think, a dozen Mohs surgeries to remove skin cancers. I had melanoma early on. And the surgeon asked, have you had a lot of sun exposure? I said, let's see, how old am I? Yeah, I've had 77 years of sun exposure. And, I had sun exposure before sunscreen was invented. You used to use baby oil and things like that to get more tan.
Yeah.
Not less. I've always been a swimmer, a surfer, a sailor, and being bald at age 21 from too much testosterone, my scalp has got exposed to a lot of sun, so that's where I get most of my tumors. But the big, other than skin cancer, which it's behavior driven, right? You go out in the sun. It's like smoking. It's we now know it's largely preventable, even though some people have a higher genetic risk. But it turns out my prostate cancer was due to a discovery we made of the testosterone, or the androgen receptor. Its expression is controlled by triplet repeats, so repeats of 3 letters of the genetic code. The more repeats you have associated with the gene, the lower the expression you have of the testosterone receptor. So men that are sterile, that doesn't matter how much testosterone they have, they're just completely sterile, have a large number of repeats. In my case, I've had the lowest number recorded of triplet repeats. So I have massive overexpression of the testosterone receptor. But endocrinology studies showed I had subclinical levels of testosterone. But what it means is because I had so much receptor being activated even by the tiniest doses, my body down-regulated testosterone production.
Interesting.
But an endocrinologist said, you have too low levels of testosterone. We have to give you exogenous testosterone because you're too low. So they didn't measure the receptor. It's like just measuring one hand clapping and you get nowhere. We cultured the cells from my 2 different tumors in the prostate. They both grew 10 times faster when you put testosterone on them without it. So I probably had very small, slow-growing tumors. And there's 2 types of prostate cancer. There's the kind you die from, and there's the kind you die with. And so it's quite possible I had the kind you die with, not from, but testosterone accelerated. It's like pouring gasoline on a fire, accelerated it to a tremendous
How long had you been on the testosterone treatment for?
I was probably on it for like 2 or 3 years. In my PSA levels kept going up, but they did biopsies. They did the old style MRI and told me that I was cancer free. But the biopsies, it's just like taking random shots at something with a dart, and they missed the tumors. I had 2 tumors and they missed them with the biopsies. So if it wasn't for the testing my own new MRI machine, I wouldn't have found it.
Wow. That's incredible. So did you also have other markers like that might have been related to your parentage and et cetera, like your father dying of sudden cardiac death? Did you also have a marker for that?
There's nothing that's very clear cut. I do have the marker for risk-taking behavior.
Well, okay. You don't need to have your gene sequenced to know that one.
Well, and also I had the testosterone, too much testosterone phenotype of being a risk taker, being aggressive, being totally bald at age 21. So I had a full, as a kid, long blonde hair, surfer head, and it all disappeared pretty quickly. So too much testosterone can be helpful, but also a disadvantage. But
Sure. So what do you think is the next big thing in this field then? Where do you think it's going to go? Maybe understanding, how long does it take for us to actually start to understand these nuances? Because it seems like you could have false positive, false negative, because we don't really understand things like the BRCA.
Well, that's why we have to put things together. And the way this developed, we talked about it a little bit yesterday. One of the genetic findings, because my genome was the first one in history to be sequenced, lots of people have analyzed my genome. And one of the major findings...
You're like literally an open book.
Yes. And it was put on the internet for people to study, trying to show that you're not at risk if other people are looking at your genome. For the most part, that's true. And in China and other places, they use it as a weapon against minority populations. So the genetic code can put people at risk if it used to track populations. But there's nothing that was found in my genome that put me at risk for losing health insurance or anything else. But the notion was of I wanted to see, so ApoE is a gene associated with Alzheimer's risk. And so obviously I was worried
Sure.
as I was starting to age that was I developing Alzheimer's? Because everybody after 50 starts to have some memory issues.
It's called senior moments.
Yes. And some of those are natural and some are progressive and with Alzheimer's. So I tried to get a brain scan and a PET, amyloid PET scan, but they wouldn't do it because I didn't have any symptoms. It's sick care.
What a paradox, because it's the doctors that have to, sign up for the Hippocratic Oath, and which is really the first axiom there is do no harm.
Yeah.
So if it's a diagnostic that might be costing money or have other associated, expenses associated with it, whether they be cost-wise or
in the dollars are what
time-wise.
became the big determinant, not the patient.
Yeah, exactly.
So my friends, head of neurology and head of radiology, agreed to do it as part of an experiment if I would do it under a pseudonym.
Really? Okay.
Because they thought if they do the test and the genetics meant something and they prove that I'm developing Alzheimer's, that would make it impossible for me to raise money for new ventures, to head up organizations, to get major grants, et cetera, because it would be viewed as a detriment.
Right.
That's why Ronald Reagan never got tested, even though it was pretty clear he had fairly severe Alzheimer's. But they called me shortly after, and they were blown away. They said, you have a brain of a 40-year-old. I was in my 60s at the time. And the amyloid PET came out totally negative. So there was no correlation at all between my genome test of being heterozygote, means one copy, that was bad, and Alzheimer's. And now we've gone on, that was my basis of starting human longevity, was clearly just the genetic data on its own was insufficient without having the corresponding phenotype data.
Ah, that makes sense.
So I set out to do the first major clinic of doing both together.
And the only way to get phenotypic kind of data would be
all these non-invasive tests.
To see if it's actually expressing the marker.
So that started, and it's like just trying a new experiment. We didn't know what the outcome would be. We thought maybe we'd just get lots of physical characteristics to find out what the genetic correlate is. And I was stunned first with starting with my own finding of having a major cancer that was likely to kill me, to finding that half of everybody that thought they were healthy had some major disease brewing in them that could be, in most cases, turned around. So that turned into this major new approach we're trying to take in medicine. But people
Which
get
is more about prevention, preventative.
totally preventative, which is what, in theory, the genetic code, the genome should, once we understand it, be extremely useful for. It is now in neonatology and pediatric disorders with known inborn errors of metabolism that if they don't get the right chemical early on, they'll die as an infant from. So it's already useful for true genetic disorders that we've, the Mendelian genetics that Victor McCusick was the hero of cataloging. But for truly understanding your and my personal risk for cancer, for heart disease, for strokes, for aneurysms, we don't know the correlates for all those in the genome because people have not been doing the physiology. They'll do genetic studies on people with disease, and that's sort of how this false information develops, like on breast cancer and on Alzheimer's disease, because there is a correlation. So people that have two mutated ApoE genes, we find about an 80% correlation. 80% of those people do have Alzheimer-related brain changes. But 80%, it's not 100%. So it means it's a risk, but it's not predictive. If you have this genetic marker, you will get Alzheimer's. And so that's why we need so much more data to get to be able to actually understand it. And that's what we're doing with trying to expand what we started, human longevity. We have a major proposal into the government to create mobile clinics to go into indigenous populations, people that can't afford the $20,000 a year that HLI charges, underserved communities, changing women's medicine. There's so many things that could be done with early detection and preventative paradigms, but the medical community is against it because they've been trained. It's unethical to do tests.
Their incentives are frankly misaligned.
People. Yeah.
The incentives are misaligned. It's like one of the things I've noticed in healthcare is that often, especially in therapeutic indications, you have a scenario where the way the compensation is done is based on cost plus.
Yeah.
So the insurance company, the hospital, the doctors themselves are incented to pay more for the cost basis of a product or treatment modality, because then they can apply 6% on top of it, ranging 3 to 6%.
And you can't get paid for saving people's lives by discovering things in healthy people.
No, that's right. So the whole system is built on the wrong premise. In this way, I think the Chinese have, the whole ancient traditional Chinese medicine thing. You pay the doctor when you're well. You stop paying him when you get sick. Then you pay him again when you get well again. I wonder sometimes if I think that's what insurance is intended to do, but I think that there's so much regulation of and misalignment of incentives that are frankly driven by lobbying efforts, and CMS as well.
So health insurance companies,
Medicare,
don't have you as a client for very long. Now, for example, at my institute, every 6 months, employees can switch and get a different health system. So in contrast, life insurance companies were very interested in our new approach.
I'm sure, yeah.
Because if
they
could
do
predict longevity, they could make billions of dollars by knowing people will live a little bit longer than they're paying for.
Or just reducing somehow the amount of end-of-life care necessary.
But they talked themselves out of doing the experiment because their rationale was maybe individuals will start getting these predictions and they'll hold off getting life insurance because they know they're going to live longer.
Oh, gosh.
I mean, so no data, but they just rationalize themselves out of it. But so health insurance companies are, they make more money the earlier you die, or what pharmaceutical companies, as you know from being head of one, they love chronic care. Diabetes is the favorite disease. Hypertension is great. People have to pay for drugs for life versus you cure something and it was gone. And so that's where you mentioned things like about gene editing and other approaches. It's early on. Ex vivo gene therapy and ex vivo gene editing means you're not doing it in the body. You're taking cells out of the body and modifying them. Are showing some real exciting things with treating cancer, possibly with other diseases, certainly blood-borne diseases, so you can take out blood cells, bone marrow cells, and make some changes.
Have you seen this new ultrasound treatment that people are talking a lot about for cancer treatment?
No.
Yeah, it's very targeted ultrasound that is being used to eradicate cancer cells, which is sounded interesting. I don't know that much about it just yet, but I do know it's getting some traction. Quite a lot.
I'll look into it because there's, we've talked about the unknown. That's what we did with the Sorcerer Expedition. We just went out and explored the unknown. Essentially, what I've done at Human Longevity, it started out to be an experiment to learn how to interpret the human genome better. And as a result, we made the discovery of half people out there wander around with diseases that they're unaware of, and we can save their lives by discovering those early and treating them early. And we don't have incidentalomas anymore. That was an artifact of X-ray. The new MRI algorithms, this restriction spectrum imaging that we've talked about that measures the water in the nucleus of tumor cells, as an example, totally changes the technology. But the medical mindset, the training is still locked in the 70s with CT scans and incidentalomas. So even when we've diagnosed, for example, renal carcinoma, it was a billionaire client. He went to Stanford to get a second opinion, and he had a standard MRI there, and they said, no, you just have cyst, that you don't have renal carcinoma. We had to call surgeons there. Finally, we got him to see somebody else at UC San Francisco that understood this new technology. They did a biopsy, and he did, in fact, have renal carcinoma, and credits us with saving his life. Standard medical care at the best institutions, aren't ready for new approaches and new information.
I had just a thought just now that's kind of interesting. I wonder if that cells that have mutated, if their hydrophilicity or hydrophobicity changes inside the nucleus.
So a lot of proteins change in cells. So one of the useful things that we do with tumor cells, we actually sequence the tumor and compare it to the individual's genome. And what we see is a fairly large number of mutated genes associated with the cancer. And so there's now a T-cell therapy where you actually take that person's mutated genes, the term for them is neoantigens, and we can convert them into a peptide or an RNA vaccine for themselves that stimulates T cells to kill the tumor. And it doesn't kill normal cells because your normal cells don't have that mutation. The mutation's only found in the tumor. So there's a lot of promising techniques. Trouble is they're expensive, they're hard to do, and with super expensive tests that are based on one-off skill sets, it's hard to get those in a high throughput fashion.
I'm just thinking now that you've identified this change in the mobility of water inside the nucleus of cells, would that be in part maybe because of a change in the acidity or the base nature of what's going on for the neoantigens? In other words, how much water is it attracting? Is that causing the change in phase transition for the water? Is it a hydrophilic? Because that, how much it attracts water versus repelling water.
Yeah, you're beyond my knowledge and capacity. I can't answer the question, but it's intriguing. What is changing it? Why is it less mobile? Why are nuclei slightly larger in tumor cells? Some tumor cells, you get polyploidy. We get a lot of copies of genome, so it's possible. We talked about just there's more DNA there and more water bound with the DNA. Maybe that's creating the gel-like structures. I'm totally speculating now, and the physical chemists out there are telling me to shut up. So quote while you're behind.
So you went on this incredible journey around the world, searching for basically all these new types of organisms that we had never discovered, and you found something like 10 to the 27th power number of new organisms, just catching them on your boat.
Yeah. So it all started, well, being a sailor and a surfer and a swimmer. I've always loved the ocean, and I'm sure there's a genetic basis for people that like water versus mountains. Some great experiments, genetic experiments were done with dogs at Berkeley, breeding Newfies with border collies. And Newfie pups, if they're exposed to water, will just, they can't resist. They go
By the way,
water.
you have the cutest dogs.
Yeah.
You definitely have the cutest dogs.
They are awesome.
They're definitely lovers.
Yeah.
One is Darwin. The other is a small little pup.
He's a miniature poodle.
Miniature
14
poodle.
years old now.
And
he
so
sailed
cute.
around the world with
He
us.
sailed with you around the world. Oh, wow. And then Rosie's only 2 years old,
Yes.
right? She's still almost a puppy.
He's 14 pounds. She's about 50 pounds. She's a golden doodle.
Yeah. But they're the same color.
Yes.
Did you genetically engineer that?
It came out randomly, or just our own selection of them. But
wow.
But they are wonderful animals, and they're lovers, not fighters.
Oh, I could definitely.
And they go to work
that.
with us all the time, so they love adventure and people. And
how fun!
They don't love flying in the airplane with me. But,
so you went all the way around the world, and you caught all of these different new organisms. I mean, you had to be way more successful than you thought you were going to be, and I'm sure a lot of people told you it wasn't going to be possible.
The very first experiment changed the entire field. So there's a paper that motivated me. It was by a fairly famous scientist who, in the Proceedings of the National Academy of Science, published this paper talking about how little life and microbial diversity there was in the oceans compared to soil. And that just didn't make any sense to me, knowing that at least half of all the oxygen we breathe is because of microorganisms in the ocean. And because the tool that we developed for sequencing genomes, the mathematics are so powerful. And I learned this early on. After we sequenced the first genome in 95, people would send us things like clinical samples. And we got a clinical sample from Norway to sequence because the patient was refractory to treatment. And when we sequenced this, in theory, the one bacterial species, the algorithm assembled two separate species. So it showed me the mathematics were far more powerful than any of the scientists and scientific ideas. It was sort of like the truth serum. If there were two species, it would find them. And that's how I learned that we could rely on the mathematics to deal with mixed populations. And so we decided to try it in the ocean, and we chose the waters off of Bermuda because that was supposedly, they described it as an ocean desert where there was very little life. So they numbered the organisms one at a time, and they were up to SAR 11, so Gaster C 11. So they really thought life was sparse there. So we just took a barrel of seawater and these tiny filters and filtered out all the microorganisms, isolated all the DNA at once, and sequenced it. And we discovered, just in the first experiment, over 1,500 new organisms.
Wow.
And discovered something 1.5 million new genes that people had never seen before. That was just the first barrel of seawater, just testing the experiment that most scientists thought would fail, that the mathematics wouldn't work or there'd be nothing there. People were at both extremes. You won't find anything or the mathematics won't work. Mathematics worked fantastically, and we found a lot. And I devised the expedition from there, following Darwin, following the Challenger expedition, that every 200 miles around the ocean sent a dredge to the bottom, because the theory in the 1870s was you couldn't have any life below 1,800 feet. What I argue is most scientific discoveries are proving the idiots of the wrong of the past wrong. I mean,
the only real obstacles are the ones we believe.
Yeah, I mean, how those ideas develop, but the Challenger found life at every depth. They discovered the Mariana Trench and changed the view of life in the oceans. It would be at every depth. So we followed their route, starting in Halifax. But what we found is every 200 miles, 80% of the sequences in the organisms were totally unique. And so that the diversity kept mounting. We got physicists and astronomers to join us because they decided we were discovering more organisms in the oceans than there were
stars.
Planets and stars in the universe. So, and the number is vastly bigger. We live on a microbial planet. We did the first sequencing of the microbiome, showing that you have about 3 pounds of your body weight are microorganisms. We are hosts for microbes, we're hosts for viruses. We live in a microbial environment. We just can't see them with our own eyes. So Darwin had no idea this world existed. In the South Pacific, it'd be crystalline water where you can see for 200 feet. It just looks like absolutely pure water. Every milliliter has 1 million bacteria and 10 million viruses. It's just not part of our visual acuity. If we could see at that level, the world would look vastly different. So what we had is now the new tool of DNA sequencing, so we can see things that nobody could see before. So before we did that experiment, the world of microbiology depended either on seeing something in a microscope or being able to grow it in culture. Most organisms in the world won't grow in culture, so if they couldn't grow them, they didn't exist.
Wow.
And so we've shown that the world was about 30 orders of magnitude more complicated than anybody imagined. And my favorite question I get on the book tour is, you showed there's all this stuff out there we didn't know anything about. What else don't we know? And that's the right question, except I can't answer it. We can only answer it by...
We don't know.
Going out and doing experiments and asking questions. When we made the first synthetic cell, the biggest discovery it took us so long to do because 25% of the genes essential for life are of unknown function. Nobody's seen them before. They hadn't studied them before. We can't get life without them. But biologists already thought they knew almost everything.
Well, physicists in the early 20th century believed that we understood all physics already.
So every time we reach this plateau of knowledge that people think we've really saturated knowledge, that's when the next level of major discoveries start to take place, because we show how foolish these ideas were. When we sequenced the human genome, the biggest discovery was that humans only had around 20,000 genes, because scientists were speculating we had 200,000 to 300,000 genes, that there must be one gene for every human trait and function. And they were just blown away. We have basically not that many more than a fruit fly, and the same as all other mammals.
It's incredibly in line with conservation of energy and information.
But, having 20,000 genes with 100 trillion cells, we have 100 trillion different combinations of those genes working. You don't need a whole lot more. The combinatorial nature of that is beyond most people's comprehension.
Have you ever looked into this question of the 23 chromosomes versus the 24, and the fusing that humans have versus primates?
So we get a lot of splits and fusing of chromosomes. Those are easily detectable now that they were detectable usually doing microscope analysis. That's how chromosome breaks and joining were found. And they're fairly frequently found in kids with genetic disorders. But now we see those by sequencing the entire genome, both sets of chromosomes that we get from each parent. When you assemble that, your genome would be totally different than mine. And the mistake that's happened in the field for the last 25 years is my genome and other genomes were used as a reference. Illumina developed this technique for sequencing very little pieces of DNA. And so instead of assembling somebody's genome and looking at it de novo, they would just see how it lined up on my genome or other reference genomes, which doesn't really tell you what your genome is. And now the techniques have improved a lot enough with single molecule sequencing, reading bigger pieces, that we can now go back to where we were 25 years ago with the first genome of actually assembling each person's genome. So we're going to be on the verge of 100-fold more discoveries from the genome, especially as we can start to compare it to people's phenotypes.
So that's kind of the next thing then.
Yeah.
I think that answers the question.
Yeah. Just genomes on their own are great for population studies.
Somehow, it's the correlation with the phenotypic expression.
It's not exactly a brilliant idea on my part of putting physiology together with genetics to understand it, but apparently NIH hasn't caught on to that yet.
Well, it's still the sick care model.
Yeah.
I think this is a big leap into a different paradigm altogether. So let's go zoom out for a moment. You also wrote a book that was faster than the speed of light, and I thought
Life at the speed of light.
Excuse me. And I thought that was really interesting because you had actually created for lack of a better term, a replicator, and patented a replicator of being able to send.
We call it a biological teleporter.
A biological teleporter. Okay, that's
a Star Trekkie.
That's a better Trekkie way to say it. That you could pass genetic information, right, and communicate it as we normally would, and then instead of having to send that actual organism, you could effectively recreate it.
Yes.
In another location.
Yeah. And this came about with one of the first major flu pandemics. I had met the mayor of Mexico City, and that's where one of the major flu pandemics started. And he called and asked if I could help by sequencing the flu virus that was killing all their citizens. And I said, yes, send me a clinical sample. Mexico couldn't send it. The U.S. wouldn't allow it to be imported. So basically it couldn't be done. So when a major new outbreak started to take place in China, it was the H7N9 variant of flu. Some forward-looking Chinese scientists actually sequenced the isolate and posted the sequence on the internet. My team downloaded the sequence, so this is the first incidence of actually teleportation, and we remade the virus in my laboratory in La Jolla, and we were the only source that the CDC and pharmaceutical companies actually had of this new strain of flu virus, because we made it. We made it in 24 hours.
Please tell me you have no contact with Wuhan.
No, and that's another story. The NIH should have never funded gain of function research in a laboratory where they have no control over it. That's high-risk research. I am totally certain COVID developed in the laboratory. 3 people were sick. Maybe they went to the market as well. But there's 2 governments that don't want to know the source, the U.S. government and the Chinese government. Because if the U.S. government, Francis Collins funded the work there, and the Chinese government doesn't want to be blamed for
I
making it in
commend.
the lab.
I do commend. The Rand Paul, who who actually went after this, and he's been here in the office. In fact, he came to visit here in the office with me the week before the lockdowns. It was 2 weeks before the lockdowns. I was in Egypt. I went to Egypt, and I made it out of Egypt with 1 day to spare before they closed the borders, and it was pretty scary. But he basically has been a super outspoken advocate and done a ton of research and has been somewhat vilified in a way for having, you could agree with his politics or not. I'm not saying whether I agree
we
with
don't
his
agree on
politics,
politics,
but he went after this issue.
A stop clock is right twice a day, as they say.
That's right.
He can be right on some things. He was right on this, in my view. And but NIH actually tried to cover up the fact that they did this funding.
Well, I mean, it was hard to cover it, though. I mean, even the publications were all saying, thank you to the NIH for giving us the gain-of-function research funding.
Yeah.
I mean, come on.
Yeah.
It was kind of funny.
But
not funny,
except
but
it's tragic.
It's tragic.
And they promoted this study of a group of, quote, synthetic biologists, saying there were no tales that the virus was synthetically made. They're looking at technology from 20 years ago. When we made the flu virus, our team made 20 variants of COVID to provide to the pharma companies to test their vaccines on. It took 2 weeks. You couldn't tell by looking at the sequence whether we made it chemically or it was naturally occurring. The technology we've made is so far beyond. And so it was a BS misleading paper to claim that there's no indication that it was synthetically made. So it was just more stuff to try and cover up what both governments did.
I could not agree with you more. And actually, there was an article that came out in January of 20 in Scientific American that someone, and you probably know the people, had actually sequenced and found that there was a relationship between two snakes. And this was very quickly hushed up. And it was an Asian asp and a king cobra that they'd put in an airborne virus. It was not a pangolin. And I have to believe that, and this was like, I can't remember, it was either Cambridge or Oxford or Harvard who'd actually done some work on sequencing this and found the relationship that it was similar to snake venom. And that's why you would have the cytokine storm. And I'll send you the article if you haven't read it. It was in Scientific American, which is a pretty reputable publication. And, the whole story, I think, is speaking to a lot of the travesty that has become, in a way, our educational system. Also the governments and the way governments are acting. Are they the friend or foe? That's the hard thing to sometimes recognize. Especially, today, I don't know if you saw the whole thing that's happened on telephones. Telephones are all out today, a very large percent.
I did see that, but they didn't say what the cause was.
Nobody knows. Nobody knows. It's just the FCC is investigating it now. But, I mean, it is kind of fishy. After we watched this film on Netflix that was called Leave the World Behind, this was the premise of the film. So, but I tend to think that
a lot of people get their brilliant ideas from
art imitates life, right?
And
and life imitates art. But I tend to think that one of the big challenges we face in the world is that we have so siloed our educational system so that we have these different disciplines and never, the twain shall meet. And yet what you've been working on has been this incredible nexus between mathematics, computer science, and molecular biology, which is what made it all happen. And, I tend to think that, as I look at the universe from a cosmological perspective, I think of, applied mathematics is geometry. Applied geometry becomes physics. And applied physics becomes biology, or applied physics becomes chemistry, Applied chemistry becomes biology. Applied biology becomes psychology. Applied psychology becomes sociology. Applied sociology becomes philosophy. And applied philosophy is back to mathematics.
Yeah.
They're all interconnected.
That's why physicists love to take claim for everything.
Yes, well, they do, but actually it'd be the mathematicians that would claim it better, maybe. But, what do you think we have to do to fix our educational system so that it's more multidisciplinary and more polymathic?
Well, first we have to get away from a rote memorization system and actually go to an education system. So every summer for several years in a row, I spoke to 20,000 high school students in a soccer stadium outside of Boston. And they were selected as the best students at every high school across the country that were interested in science and medicine. And I was explaining aphantasia to them and photographic memories. And I said, my hypothesis is the education system is a giant genetic selection system for photographic memories. And I said, you're the top students in your high schools. I'd like a show of hands. How many of you and how many of your parents think you have photographic memories? It was 99% of the audience put their hands up. It's a perfect example of genetic selection. Here, the top of their class. How'd they get to be the top? They have photographic memories. They're great at rote memorization. There were maybe 20 people there after I explained what aphantasia was that I can't see pictures that thought they had aphantasia. But you have to be brighter in a different system to survive against the photographic memory people to get there in the same way. And the original studies on aphantasia were from the late 1800s. And they said it's a trait primarily found in scientists.
Wow.
And those studies were repeated recently, and it was still sort of true, but, sort of a selective audience. I think a lot of artists, there's so many brain types, and that's where genetics, genomics mixed with the MRI resolution, we should be able to see an exact pattern associated with photographic memories versus aphantasia versus people.
We're not very good
in
science of identifying patterns that are cross-discipline as well.
Well, the government doesn't want to fund anything associated with measuring intelligence in people.
I mean,
because the fear is that it will be used as a discriminatory tool, even though it's
well, you did see Yale just announced yesterday that they're back to SAT.
Yes.
They're back to standard tests.
I was very surprised to see that. I didn't read their rationale, but I did see that headline. And
I think it's because it wasn't working. That's probably it. Dartmouth did the same.
And that also with the new rules that they can't do affirmative action and anything to bend rules for admission to help people in disadvantaged classes, you might as well go to a straight numerical system.
But all of these systems of affirmative action, et cetera, they are inherently racist.
Yeah.
And I have
well, as they've shown that IQ tests are, because everything is socially developed. And so, but even assuming there's such a thing that can be measured as IQ when there's at least 20, if not 100 different types of intelligence. There's, I have what's called street smarts because I go on intuition and understanding because I can't see pictures to memorize things. And you told me you see the world in pictures. And,
I'm one of those automatons, I guess, that the world is kicking out right now of visual memory.
It's a fantastic tool. I had a nice discussion with my book and, talking about memory types and imagination. And the person I was talking to sees the world totally through his visual memory.
Yeah.
And that's how he classifies everything. It doesn't mean people are less intelligent. If anything, it's higher intelligence if you can use that as a tool to do something unique and better with it versus using it to skate through school because you didn't have to work to memorize stuff.
No, it's funny. I learned 8 languages during my lifetime, living in 9 different countries.
I've had trouble learning one. So
well, probably the visual. For me, I can literally remember the page numbers of books that I studied when I was 19 years old and where to find the exact reference. So it's a weird thing. But I used the photographic memory in order to identify patterns. that I would see across many different areas, that very broad spectrum of things. And that's what ended up becoming the basis of a lot of my intellectual property and patents and such. It was
By the way,
mainly
I
patterns.
do that by looking at graphs and looking at pictures. So I'm very visually oriented, and I would not do well in living in a dark cave or a prison cell. I've tried to avoid prison as much as I can.
That's good.
So I'm actually very visually stimulated, and looking at graphs of information, I get a lot of insight and understanding.
So you can track the correlations of things.
You can do it in your head. I actually have to see the graph and see the picture.
That's really fascinating. I think that some sort of wholesale change, I think going back to standard test is, among other things, is not the only thing that people should look at, but I do think that there's merit in it, because what we've experienced with society recently is a breakdown in merit. It's the meritocracy that has been established has been, in a way, vilified. And I think it's happening all the way through history, references, and everything. I just saw yesterday that the new Google AI, you can't even, I saw a post yesterday on TikTok of a black person who was basically complaining that he kept trying to make a white family on the Google AI, and it wouldn't allow him to. It literally wouldn't allow him to. And he's like, I'm an artist. I want to be. It had to have genetic diversity. So, and it had to be, race diversity in particular. So I think that and it actually had a warning thing on there too that would say, we don't do things that are racially unfair. But now there's no white people. It was like unbelievable. So to me, it's kind of like gone to the imp of the, it's gone too far to a certain extent. But I hope that our educational system will move more to a balanced perspective and value being able to see patterns rather than just calling it pareidolia.
Well, it used to be, at least in some level, maybe just in higher levels of education, evidence-based decision-making. Our whole country has gone away from that. They'll take the word of some crackpot over 100 years of science.
But maybe that's also part of what's happened with us knowing at a cellular level almost that the government was not being truthful about COVID.
Yeah.
I think it exacerbated that whole thing where people are now very distrusting of government. People, if you follow the government, you would have had 12 vaccines now, 12 boosters.
Well, vaccines have saved tens of millions of lives, and the anti-vaxxers are putting us all at risk by reintroducing mumps and measles and all the things that we thought were gone. But it's healthy to not trust the government. It's healthy not trust what comes out of pharmaceutical companies. When profit is a motivation, you have to always question the motivation of what people are saying, even though pharmaceutical companies totally exist only by doing something good for the population, by making drugs that treat disease. But they do that so they can make billions of dollars. It's not altruism. Now, a lot of people, they're doing the work, and the scientists are doing it for altruistic reasons. They don't get paid billions of dollars, but it's a complicated system. And certainly with this election coming up, there's so much misinformation. One party's thriving on it. That's why I worry the most about the AI tools, particularly ChatGPT, which by definition comes up with BS or science fiction, a third or more of the time.
Oh yeah, it's true. I've tested it myself.
And so people want to use this for doing medical diagnostics. And I said, well, only if you can sue your computer for malpractice. And some of the stuff is so convincing real that, we're going to need a whole technology to, help discern truth from things we see in videos. It's tough on Instagram now. It's tough. I get targeted by so many places with phony email, people trying to hack into our genetic computers and things like that, that on first glance, they look real. They look like they're from my bank. They look like even from my own HR department. And you just have to always look at the whole email address, where it came from, to see how much phony targeting there is out there.
Oh, there's no doubt about that. So my final question to you, and thank you so much for being here with me, and also for people that want to follow your work and learn more about you, where could people go?
Well, jcvi.org is our website for our not-for-profit institute. Amazon selling all three of my books: The Life Decoded, about sequencing the human genome; Life at the Speed of Light, about making synthetic life; and now the Voyage of Sorcerer 2, about discovering all this life, microbial life in the world. Those are great places to start.
What is it that you hope that, you've certainly had the opportunity to leave a big impact, because your efforts and your work and the teams you've led and everything has culminated in changes in how we perceive ourselves. What would you like to be remembered for the most?
The work that I'm about to do.
Great answer.
So it's, I did an interview, again, for Nature magazine, and I said, I consider retirement as the equivalent of death. In fact, that's what it is in reality. A high percentage of people die within 3 to 5 years of retirement.
Yeah.
My father died 3 years before retirement, but people lose their identity, their motivation, their so many aspects. Being a scientist is one of the most privileged positions that we could invent for humankind. I get paid to ask questions about life, to make discoveries. It's just a wonderful existence to do that, and knowing that you're affecting people's lives, that our methods are saving lives every day. It's incredibly motivating. I'm hoping human longevity helps me get through the next 20 years of trying to take this new medical paradigm out to see if we can change more of society with it.
The feeling I had after leaving your office yesterday was looking at all that we talked about and all that was in your office and this incredible career you've had. Is, for myself, I was very inspired, and I felt the best is yet to come.
Well, as you know, with all the you've done so many different things. Our lives are the mathematical sum or the exponential sum of all those things. So everything I've done in my life up to today is brought me to this next phase that we're trying to do to have a much bigger impact on medicine with pre-symptomatic, non-invasive testing coupled with the human genome.
Thank you so much for all that you've done and for bringing your light to the world and to science and humanity. And I very much appreciate you taking the time with me today.
I enjoyed it thoroughly, and same with our discussion yesterday. So thanks for including me.
Thank you. We'll see you next time.
Yes.